Hello, my name is Zachary Grunewald, and I'm a doctoral candidate at the University of Missouri, and here I will summarize my dissertation research titled The Role of TRAF3-IP2 in Obesity-Associated Vascular Insulin Resistance and Dysfunction. My doctoral advisor was Dr. Jaume Padilla. Cardiovascular disease is the leading cause of mortality worldwide. Recent statistics published by the American Heart Association identifies cardiovascular disease as the highest incidence of death worldwide. Importantly, obesity ranks among the second highest independent risk factor for the development of cardiovascular disease. Statistics published by the Center for Disease Control and Prevention in 2020 indicated that approximately 42% of the United States population is obese. Obesity is defined as a body mass index greater than or equal to 30. Notably, obesity is associated with a variety of metabolic and vascular derangements, including chronic inflammation, impaired vascular relaxation, hypertension and arterial stiffening, microvascular dysfunction, as well as impaired glucose control and insulin resistance. Importantly, despite the known metabolic and cardiovascular derangements associated with obesity, the mechanisms driving obesity-associated vascular dysfunction remains poorly understood. Thus, we hypothesize that TRAF3-IP2 mediates obesity-associated vascular dysfunction. Importantly, TRAF3-IP2 is a known master regulator of inflammation and cellular stress. However, it remains unknown whether TRAF3-IP2 mediates obesity-associated impairments in glucose tolerance and insulin sensitivity, elevations in blood pressure and arterial stiffness, impairments in microvascular function, as well as obesity-associated endothelial dysfunction. Therefore, my dissertation work sought to address these questions. My dissertation project was broken down into two aims. In Aim 1, we sought to determine whether elevated levels of TRAF3-IP2 can cause vascular insulin resistance and vascular dysfunction. In doing so, we overexpressed TRAF3-IP2 in cultured endothelial cells as well as isolated arteries from mice. In my second aim, we sought to determine the role of TRAF3-IP2 in a genetically modified mouse model where TRAF3-IP2 is absent. We compared the effects of the loss of TRAF3-IP2 to wild-type mice that contain TRAF3-IP2. Furthermore, these mice were fed either a high-fat diet or a normal chow diet for 16 weeks. And lastly, this study was also conducted in both male and female mice. From my dissertation experiments, we found that increasing TRAF3-IP2 in vascular tissues causes vascular insulin resistance as well as dysfunction. Next, we found that the loss of TRAF3-IP2 protects male mice against obesity-associated vascular disease. Namely, we found that when TRAF3-IP2 was genetically ablated from obese male mice, this protected them against elevations in blood pressure, arterial stiffness, endothelial dysfunction, microvascular dysfunction, as well as glucose intolerance. Lastly, we found some interesting sex differences between males and females, in that we found that females had lower expression levels of TRAF3-IP2. Importantly, this low level of TRAF3-IP2 expression coincided with protection against obesity-associated vascular impairments in females. Taken together, we concluded that TRAF3-IP2 plays a causal role in the development of vascular dysfunction in the setting of obesity. This concludes my dissertation summary.